Well, we will move on to our next speaker. Our next speaker is Dr. Diane Rowland. Diane is the chair of the agronomy department and the professor of crop physiology in the Institute of Food and Agricultural Sciences at the University of Georgia. She also directs the Center for Stress Resilient Agriculture, where she specializes in physiological mechanisms of crop stress, particularly related to water deficit and its impact on root structure and function. So Diane, you can share your screen and I will turn control over to you.
Can you see my screen?
Yes.
Okay. Great.
There you go.
Well, good afternoon. I want to say first, thank you so much for including me in this symposium. I have to say that APRES has really had a profound impact on my career as a peanut researcher, so it's wonderful to be a part of the group and to address such an important problem as aflatoxin. So I need to qualify my talk today to basically say I'm going to give a 30,000-foot view of an approach that our team has been working on over the last 7 years for not only addressing crop stress, which is close to my heart as a physiologist, but also to build in ways of assessing aflatoxin. There are a couple of core points to this approach that we hold dear to, I guess. Essentially, the first is that we think it's important that assessing aflatoxin, and I was really excited to hear the points of Marshall, it has to occur across the entire system. So to be able to just pinpoint, say, pre-harvest aflatoxin production is not really the best approach. We need to start to analyze the risk across the entire system. The second core to our approach is making sure that we take a transdisciplinary approach. So we have a bit of semantics here. We talk a lot about multidisciplinary, interdisciplinary research, but how our team defines transdisciplinary is a way in which we truly embed within each other's programs. So we have to learn each other's disciplinary language. We then have to formulate the problem and determine what the problem is that we want to research. We have to develop the methods together. We then have to collect the data together and ultimately analyze and interpret the data together. So this takes a lot of time, it takes a lot of effort, but I think by taking this approach, it has brought us success in dealing with aflatoxin. So really what I want to present today is where we are in a project that was funded by USDA NIFA in 2018, in which we've kind of taken these philosophies and applied them to the research of aflatoxin. So this is a diagram that was actually in our proposal, and it illustrates then where we want to take this kind of systems approach in our project. So it's a very simplified schematic of the system and identifying the vulnerability points to aflatoxin along that system. So for example, at the beginning, where we begin with planting the crop and the stress events that are experienced by that crop, even though we're not in the late season. As a physiologist, I understand that those may make the crop more vulnerable than to the late season stress events that we know are so critical for producing aflatoxin and make the crop so susceptible. So we want to be able to evaluate stress events across the entire life cycle of the crop. Then at the buying point, we need to be able to sense and to assess then the vulnerability, the aflatoxin, the fungal, the fungus that is present within the harvested samples that are coming to the buying point, and then eventually moving into the warehouse and storage, and then during the shelling process. So being able to assess those vulnerability points all along the system is really the critical approach to looking at this problem. In the light green are the current, in most cases, the current assessment processes that are in place. And so, for example, we have those at the buying point and then during shelling. And in most cases, these assessment points, as Marshall pointed out, are either low tech, where we're looking at visual mold, for example, the presence of visual mold, or they're a single approach. So there may be a chemical test for aflatoxin itself. What we proposed in this project was to actually add other assessment points, and those are shown in these dark green boxes. And they combine an approach of advanced sensing technologies that include imaging, and they include VOC, or volatile organic compound sensing, for the fungus, for aflatoxin, and for crop stress itself. And then finally, to integrate the data across the system using more advanced analytical approaches, both looking at maybe the traditional modeling approaches, for example, with DSAT that we've used before, but then also moving into ways using other advanced modeling approaches, machine learning, and hopefully eventually artificial intelligence. So this kind of sums up the methodology for our approach in the current project. So again, that includes proposed assessment points that use advanced technology, using integration of those sensing and data across the system through advanced data analysis, which hopefully then produces a model that we can use for risk assessment throughout the system at those different vulnerability points. So in general, we've taken imaging processes that involve thermal imaging. Thermal, we know, is really critical for determining crop stress because it's related to canopy temperature, which is related to transpiration. So thermal imaging has been a large part, as well as hyperspectral imaging. So the ability to identify the spectra that are associated with aspergillus and with aflatoxin is one of the goals that we're after. We've also used the imaging on different platforms. So you can see some pictures here in the field where we're looking at thermal or hyperspectral images of the crop itself, and then bringing the imaging processes to pods and to kernels. These are coupled with VOC detection, so volatile organic compound detection, and again on plants and pods. You can see a picture of our team in a very small rainout shelter where we're looking at VOCs that are emitted from a small plot of drought stress plants, to VOCs collected off of a field, and then VOCs collected off of shelled kernels. Then taking that data from these advanced sensor sources, we've been looking at the DSAT aflatoxin modules developed by Dr. Booty, also Bernoulli hierarchical models, and then moving into machine learning, and again, hopefully to artificial intelligence approaches. Just to review, VOCs or BVOCs are biogenic volatile organic compounds, and they're basically low molecular weight hydrophilic molecules that are given off of various organs of the plant, including the leaves, flowers, roots, as well as the pods and the kernels. So on average, a plant is going to basically produce 1,700 of these volatile compounds. So I'm going to step you through just very briefly this project, which I hope illustrates, again, the approach that we think is going to be successful in tackling aflatoxin. So we'll start at the beginning of the chain in the field. And in this case, it's really critical to be able to detect stress in the crop. So we know stress events are either useful for priming the crop so that it's more resistant to developing aflatoxin later in the season, or to actually detect late season drought stress, which we know is critical for the development of aflatoxin pre-harvest. Those sensing data then can be used to develop risk prediction models. And so that is a key component of the eventual models that we hope to produce. So this is work by Shannon McCamus, who was a master's student in my program. And when Shannon and I discussed the best approach, we debated about whether to start in a controlled environment or in the field. I always love to go straight into the field, but we discussed that it probably would be better to start under controlled conditions in a greenhouse. So Shannon designed this unique system in which we could isolate the root soil. So those are in the big PVC tall columns. And so we could control the soil water and the stress level within that column separately from the pegging soil. And so you can see the pegging pans then above the root column. And those pans were important for not only controlling the pegging environment, but to also be able to specifically inoculate with aspergillus. so that we could control and standardize the amount of inoculation in each of those pegging pans. We tested 3 irrigation treatments to look at a range of drought stress, so 100% to 66 to 33%, and then inoculated and looked at the progression of aflatoxin. So I know Shannon presented on this last year, so I'm just going to give you a brief overview. We started with the DSAT aflatoxin module and tried to assess the components that are used in that module. Within that module, of course, soil temperature is critical, soil water status, as well as some sort of a quantification of plant water stress, usually associated with evapotranspiration, and then the cohort aflatoxin mass. So the cohorts of pods that are produced over time, that mass of aflatoxin is related to their cohort structure. So it was important for Shannon, she wanted to be able to assess that cohort structure and determine what the aflatoxin contamination progression was within those cohorts. So she individually tagged pegs on a weekly basis and then followed those pods, ultimately harvesting them and evaluating their aflatoxin concentration. And then she collected the data from the environment, soil and water temperature, the pegging, soil conditions, as well as evapotranspiration in the greenhouse. We also started with our imaging process here. In this case, we chose thermal images, and you can see a rail system that we've set up and have a thermal camera set on top that can take overhead pictures then of the plants and the pans. Obviously, thermal images we were hoping could help us assess stress level of the plants themselves, but also as we inoculated the pans, could we pick up a thermal signature associated with aspergillus and the invasion of aspergillus in the pods and the eventual production of aflatoxin. That would be awesome, that'd be an ultimate goal, but in this way we could take these images over time to assess if that was present. We also took VOC collections, and this is a system designed by the team at Georgia Tech. They used absorbent material, basically called twisters, and so these were placed on leaves using a small magnet underneath, and so it collected and absorbed the VOCs that were coming off of the leaves. In this case, we collected for an hour in this experiment, but we've been testing various ranges of time for VOC collection. Those twisters are then taken into the lab and analyzed for VOC signatures using gas chromatograph, mass spectrometry, and so you can look at the peaks of the different compounds and then use those signatures in our statistical analysis approaches. And so basically determining which compounds differed in our treatments. So there are several findings, but I'm just going to go over this very briefly. There were 3 pretty important findings from this experiment that helped guide then our subsequent field research. First was, do those cohorts matter based on our data? In this case, aflatoxin was the same across cohorts. So the tagged pegs going in 1 week, the aflatoxin contamination didn't matter over time. So one thing that was interesting to me is that in looking at the development of those pegs, so even though pegs entered the soil at the same time, the pods that developed actually had a range of maturities. So within a cohort, there was not a cohesive maturity level, and I think ultimately that's why our cohorts, it didn't matter for their aflatoxin. Basically, the cohorts developed aflatoxin likely related to the individual pod maturities. The second, when we pooled all of the environmental data and looked at their relationship to aflatoxin, we found, of course, that the maximum soil temperature, the pod zone soil water was important, had a very important and critical relationship to aflatoxin. But the other thing that was good, I think, and novel is that the water uptake of the plant at 30 and 40 centimeter depths also was important and related to aflatoxin. This is key because I think it lends itself to a breeding effort for resistance in cultivars. that maybe it's important that we look at root system architectures and function, and maybe having a root system that's able to access certain depths of water, especially during drought events, adds to aflatoxin resistance. The third point being that for our VOC analysis, we were able to separate 100% and 33% irrigation, so the VOCs were picking up a signature of crop stress. So that was our primary goal. From there, we took the knowledge that we gained from that controlled experiment and moved to the field. And so this is the part of the project that we started last year. It's ongoing now, so I only have preliminary data to discuss, but I think it's important to show, again, illustrate the approaches that we've taken. In the field, we still wanted to be able to control for the inoculation, and so we took the general idea of Shannon's pegging pans and then inserted these into the field. So these are enclosed pans that we planted then in between, and the plants then were able to peg into those pans. With the enclosure of those pans, we were able then to inoculate with treatments. And so we inoculated with aspergillus as we did in the greenhouse, but we also used Aflagard. And the reason being that we wanted to try to understand if toxigenic and atoxigenic fungi had a different VOC signature, a different thermal signature. This could be really critical for remote sensing, for example, of populations of fungi in a field, and how risky is the population of fungus in that field, for example. I know that's a long way off, but getting this kind of data, I think, adds to those possibilities. We used rainout shelter then to induce late season drought stress to ensure that we had aflatoxin production then in the field. We again took imaging approaches, in this case both thermal and hyperspectral images of the crop itself. So here are some pictures of the thermal images, and this is on a stationary platform, basically kind of emulating the possibility of attaching a thermal camera to a tractor or a piece of equipment, or even to a center pivot. And then we also use drone platforms, in this case for our hyperspectral imaging, where we could take images at the field level as a whole. We collected VOCs very similarly to the way that we did in the greenhouse. So again, attaching twisters to the leaves within the pegging pans. And this is very preliminary data. Again, not a lot of data points, and we're continuing this research this year. But so far, we have been able to detect differences coming off of the leaves in the VOC signatures from our 3 inoculation treatments, from Aflagard, aspergillus, and then non-inoculated control. The pans also allowed us then to harvest the pods and to take VOCs off of pods, intact pods, and then shelled kernels. All right, so those approaches give us kind of cover the field aspect. The next step was to move through the system and go to the buying point in the warehouse. So what's critical at those particular points? In this case, it's really important to be able to sense the fungus. So, what level of fungus do we have? What is it? Who is it? And the aflatoxin production. This allows us eventually to make segregation decisions then by looking at risk prediction and possibly combining those with the data from the field so that we know the risk coming out of the field and going into the buying point. So essentially, we're looking for aflatoxin footprints. This was important to do both with imaging and with VOCs, and then take that data into modeling approaches. And we are starting to look at hierarchical models and then also machine learning processes. Machine learning is important for target identification. So how do we identify aflatoxin or aspergillus within these images, but also for integrating the data across the VOCs collected. So VOCs, like the field VOCs coming off of the leaves, for in-shell samples coming out of those pegging pans, and for shelled samples, the VOCs so far showing discrimination amongst our 3 treatments. So that means the VOCs coming off of the leaves, the VOCs coming off of intact pods, and VOCs coming off of shelled samples are showing different VOC signatures. It's not too surprising that Aspergillus and Aflatoxin would differ, because when you look at the compounds that are within those VOCs that are coming off of those particular samples, we do see many different compounds that differ between those 2. On the imaging side, we took an approach using machine learning called MT-MIACE. So here is where, if you have specific questions, I'm afraid I can't help you a whole lot, but I can guide you to our team that's working on this approach. But basically, this is a machine learning approach that's been used for facial recognition. So facial recognition, you're basically looking for targets of features within the face that help you differentiate between 2 faces. Well, this is really similar to the problem of aflatoxin. We have these hot spots. Some samples, they've got hot kernels in them, some don't. And so to be able to identify those targets within, say, a sample of either pods or kernels is important. So we took the hyperspectral imaging approach, and here's an example of a visible image of a group of kernels, and then going and taking a hyperspectral image, and that grayscale reflectance then can be separated out into the individual spectra. So this approach, this MT-MIACE, is really good at finding targets with high variance. Again, a perfect situation for aflatoxin. So far, again, preliminary data, we've been able to identify at least 2 spectra that are associated with aflatoxin contaminated kernels that differ from normal non-contaminated peanuts. So this shows a promising approach, not only from imaging, but also from data analysis and identifying targets using machine learning that may be very promising at this point in the process. We also can imagine that these same kinds of imaging approaches and analysis then could be used for the product that's coming out during the shelling process. So if it's able to assess it going in, should be able to assess it going out. So I'm going to finish by describing our utopian vision of what this might look like in the future, how we would integrate across the system for sensing and for sensing aflatoxin, but also integrating the data across all these different points. So I'm going back to our diagram, and we have various stop points where hopefully we could mitigate the development or remove aflatoxin contamination from the chain. So starting in the season, it would be ideal to have an integrated sensor network for sensors that communicate with each other about crop stress. And so we're able to sense stress in a very complex way, maybe learn using artificial intelligence ways in which we can improve the sensing of that stress, and then allow growers to mitigate the development of stress events. Late season, we know that same sensing is really critical. In this case, we would be wanting to sense very carefully the development of pre-harvest aflatoxin to either segregate, harvest, and basically put a stop, prevent contamination to enter into the next steps of the chain. At the buying point, we would want to be able to very carefully assess the presence of the fungus or aflatoxin itself, again, to be able to segregate, to be able to cull samples, and of course that same sort of sensing, advanced approaches, and integration across the data in the warehouse. In this case, you could imagine that hotspots within the warehouse could be sensed, could be identified. Maybe someday we have robots that go in there and in the pile and they're taking out, they're culling samples. That would be awesome. That's a long ways away. But at least identifying hotspots within the warehouse, whereas you're unloading the warehouse, you can segregate and cull those samples. And then basically during the shelling process, having some integrated sensing process in which you use imaging, VOCs, other technologies that get developed to actually assess the aflatoxin during the shelling process. So with that, that's our vision. Again, I appreciate this opportunity to talk about what we're doing, be involved in the discussion, and if we have time, I'll be happy to answer questions, or you can contact me later.
Okay, Diane, I think we have time for one question. I'll read it here. Do you think that the MT-MIACE technique can be scaled up to detect aflatoxin, not aflavus, or damage at the processing speed similar to an electronic color sorter in the shelling blanching plants?
So I do, but I'm sure our team, our machine learning team, has a much better vision of how long that would take and what that process would take. My understanding is that by using machine learning and artificial intelligence, you would be able to, if you were using hyperspectral analysis, for example, or even VOC, you'd be able to eventually dial down and find the specific signatures, the image signatures, the VOC signatures that you were really after, and then the processing time is much quicker.
Okay, great. Well, thank you, Diane. Fascinating talk. So I missed one, jumping back to Diane. Do you think we need mitigation strategies that specifically target prevention of infection, contamination, and aflatoxin?
Absolutely. So I think with the sensing technologies that I talked about, that goes hand in hand then with the development of mitigation strategies. And so as Marshall's indicated, this is a multi-step, multi-time process. And I think we need to make sure that we're pairing both the ways of evaluating and sensing aflatoxin with methods that can mitigate it at every stop along that chain.
Okay.